All right, everybody say hi. That was kind of lame. Jeez. Whatever. What I'm really afraid of, and I think I did that a little bit on Wednesday for the micro class after this class, is I turned it off, and then I turned it on when I was done. And the only video I ended up making was about a 7-minute video of me looking at like the PowerPoints for the next couple of lectures or something, and just silently, and then I come up and turn it off, and like, oh shit. So, fortunately for the micro, I had a lecture from the fall that was pretty much, oh, that starts and stops right where we should. Okay, I'll just post that. I guess what's happening then is that I'm going to have this YouTube collection of me, and that I can teach molecular and cell biology in perpetuity on one hand, or it's just like, I really don't exist. I'm just an AI. I'm actually a hologram. No. All right, so we're finishing up our macromolecules. And hopefully you're kind of working on developing more or less a table chart thingamabob where you have the different macromolecules, the monomers that make them, the things that make them interesting, and examples, stuff like that. Yeah. So we've gone through our carbohydrates, sugars. We've gone through our fatty acids, nonpolar class of molecules, except, of course, we've got the specialized diglyceride with the phosphate on the third carbon, that is the phospholipid, which is got an extremely polar end to it. Talked a little bit about soaps last time. And then we got into proteins, arguably the most important discussion point that we'll make for the entire class, because we invoke these kinds of ideas of secondary and tertiary structure and the changes for everything that we're going to talk about from here on out. Everything from the way enzymes work, but also to how proteins interact with each other. Everything is dependent on that. And just to reiterate, we have primary structure is the amino acid sequence, held together by covalent bonds, very tough. Unless we do some kind of enzymatic wizardry, you can't touch the primary structure. Secondary structure is the local interactions, and so we can generate these things like the alpha helix or the beta pleated sheet, or sometimes what's just known as an acidic blob, where you got a lot of negatively charged amino acids and they just kind of stuck together. But they're held together by hydrogen bonds, charge interactions, not polar interactions. What the official biochemist term is the weak-ass bonds. And then we got tertiary structure, which is where we take these secondary structures and then begin folding them up and making a large protein. And this is how proteins work, by being able to form up into very large structures. And not to be, so the tertiary structure is also held together by weak-ass bonds. So just to finish it up in terms of structure, I mentioned this at the very end, there is what's called the quaternary structure. And pretty much all proteins exist with other protein interactions. And so you have, we know as protein interactions. And very critical, no doubt about it. Lots of proteins end up being multi-subunit things. Just as an example, the globin protein that holds oxygen in our blood is in fact 4 globin proteins linked together. Pretty much every major molecule or major protein is made up of multiple subunits, and this would be referred to as the quaternary structure. Now, it's also held together primarily by interactions, distinct weak bonds. There are a couple of little covalent bonds we'll throw in just a moment. But from a molecular biology perspective, I refuse to talk about quaternary structure other than what I just said, in that you can't predict it from the genomic sequence. And so I enjoy my DNA sequencing, and by looking at the DNA sequence, we can infer an amino acid sequence. By knowing the amino acid sequence, we can then begin to computationally infer what the secondary structure and tertiary structure is. And a couple of points about that are true nerd points, but still, the secondary and tertiary structure fold up along thermodynamic lines. What is the least amount of energy that the molecule can hold and that's how it's going to fold up. And so there are some folds that are just not thermodynamically particularly stable. Now, I was sort of naively thinking, back when I first started thinking about this stuff, that, oh, there is one tertiary structure. Okay. But it turns out that within a given, particularly these really complicated couple of thousand amino acids or whatever, there are a number of equivalent thermodynamically stable structures that a protein can fold up in. But only one or so is in fact the functional way. And as I've been kind of thinking about it and looking at protein structures and things, there are subtle changes that you could be this way, you could be that way, you could be flopped a little bit, but so the thermodynamics is pretty much all the same. And probably the part of the protein that's actually doing something, either the active site or binding domains, things like that, are pretty much always the same structure. And there's some subtle differences. But even still, there are some proteins that have a couple of different forms and end up maybe being non-functional in one shape and functional in the other. And there are a whole host of proteins that we'll be talking about later, just to throw out, that are called chaperones. And these are proteins that help to fold other proteins in the cell. So protein folding is a really big deal. Let's see. I'm starting to get excited again, so I have to take my sweater off. Because I love talking protein structure. All right. A few odds and ends, and some of this actually is, we devote chapter 4 to this, but I think it fits in well now. In that we talked about, these different kinds of bonds and interactions. There's another bond phenomenon in proteins that is worth mentioning, and it is the following. Something called a disulfide bridge. And it occurs between two sulfur-containing amino acids. There are two sulfur-containing amino acids. There is methionine, and then there is cysteine. And cysteine has the ability to do this. So you have a cysteine, and I ain't gonna draw the structure, but you form what's called a disulfide bond, which is you got an amino acid here, there's a sulfur, you have another amino acid polymer here, here's a sulfur, and you can form a disulfide bridge, and essentially covalently linking two proteins. And so not all, but many protein subunit complexes, the quaternary structure, can have sulfur-sulfur or disulfide bonds in them. And this, again, is a covalent bond, so it's pretty tough. An example of some of the interesting things is that you can have two different proteins that are disulfide bonded. Turns out that insulin actually is synthesized in the body as a single protein, but there's two cysteines that form a disulfide bridge, and then the protein as it's being processed is proteolytically cleaved to generate two subunits. The big protein complex of the immune system, the antibody, has got a lot of disulfide bonds in it as well. And we can break these disulfide bonds using chemicals that are collectively known as reducing agents. And these are agents. These are able to actually break that sulfur-sulfur bond. A couple of the ones, and just you may see these in the lab or you may hear about them. There's one that is called beta-mercaptoethanol, also known as BME. There's another reducing agent called dithiothriotol. And this is abbreviated, by the way, as DTT. So if you ever see these in recipes and things, you'll know what they are. BME smells a lot like natural gas, rotten eggs, really nasty smell. Dithiothriotol smells kind of like funky old tuna fish. And you would know this smell if you go into a hair salon that does perms. Because if you ever had your hair either straightened or curled, you know there's that funky smell they apply, or the compound that has that odd smell, and often, they mask it with perfumes, but there's definitely this sort of a funky, rotten egg, weird, fishy smell. That is reducing agents because there are disulfide bonds that are part of the curl. And so you can break the disulfide bonds and straighten the hair out, and then you rinse it out, reform the disulfides in a slightly different configuration, and you can straighten your hair, or you can put a perm in and color it. So I just thought I'd throw that in there. So, okay. We've got proteins and protein structure. We'll talk more about protein structure as we continue on, but it's something to think about. And a really critical sort of term we use is this idea of denaturation. Denaturation we can define as a loss of secondary and tertiary structure. So cooking is an obvious example of where we're denaturing proteins. I mean, that's really any kind of cooking technique you can think of is denaturing proteins. And, the easiest way to cook stuff, of course, is heat. And, of course, heat is breaking those weak hydrogen bonds. But there are other ways you can denature proteins by adding a whole bunch of salt, by adding acid, by adding base. These are things that will also affect all these weak bonds and cause proteins to unfold. And so, not to belabor the point, but first off, this is what cooking is. Cooking is denaturing stuff. Additionally, sterilizing things. You sterilize stuff by denaturing the proteins of the organism with which you wish to kill. And so denaturation is an absolute straight line to death. And all the kinds of things we think of as sterilizing agents, they're pretty much all protein denaturants. I mean, one example different would be ultraviolet light is a really good sterilizing agent. And that's not so much denaturing proteins as it is shattering DNA in organisms. So you can have things that are quite sterile, that is, no living stuff on there, but they're not necessarily clean or sanitary. I mean, there's some bacteria that even once you kill them, they can still have lots of protein toxins and things around them. And that was the last bit to talk about with proteins. All right, so our final class of macromolecules, and when I'm doing my non-majors, biochemistry of food, a bit of fuzz, pardon me. We don't get into this fourth macromolecule class, but for us, it's critical, and that is the nucleic acid class of macromolecules. Now, this guy follows beautifully in the monomer-polymer model. The monomer is the nucleotide. And the polymer is things like DNA and RNA. All right, so again, structure, there's a few critical points, but we'll save it for biochemistry that you have to memorize the structure of nucleic acids entirely. That said, there are a couple of key points that we want to throw in. So in genetics, on the street, wherever you learn your biochemistry, hopefully you've heard of a nucleic acid. And it is a 5-carbon sugar base. So there's the oxygen link. And then we've got I said the 5-carbon sugar base. I was thinking of it as the base structure. There's the 5-carbon sugar, and then there is a nitrogen-containing base out here. And this is where the nucleotides get their specific names and things. And then we, of course, have the possibility of adenine, guanine, cytosine and thymine. And of course, in RNA, the thymine is modified such that it's now called uracil. I never really worried about any of that. And yes, at one point for a molecular genetics class, I did memorize the structure. That's an E. I'm not sure what the hell I'm doing there. I memorized the structure of all four and then promptly forgot them by lunch. No need for you to memorize those. What is important, though, is the way we can think about their interactions. First, adenine and guanine, these are known and based on what's called the purine structure. And cytosine and thymine uracil, they're based on the pyrimidine structure. So we've got purines and pyrimidines. Personally, I always remember the purines as adenine, guanine. It's the same letters as my brother Andy Galuski, my oldest brother Andy, and I always think of him as being a reasonably pure person. And there are folks that are in medical school that still remember adenine, guanine this way. So feel free to use that. Adenine, guanine being the purines, thymine, cytosine, the pyrimidines. Also importantly is the way they can base pair. The bases themselves can interact in that we have AT base pairs and GC base pairs. And they're held together by 2 hydrogen bonds or 3 hydrogen bonds, respectively. Consequently, GC base pairs are harder to break apart than AT base pairs. And consequently, we will see anytime there is a naturally occurring spot that has to actually unwind and open up, it'll always be very AT rich. It's just thermodynamically easier to open up double-stranded DNA if it's AT rather than GC. All right, so there's the base. Then we've got the phosphates up here. So, and I can never remember if it was 3 or 4. I don't care, and you shouldn't care either in terms of the oxygens. We got the terminal triphosphates. And these are going to be part of a couple of things. This terminal phosphate here is used as a phosphate donor for energy requiring reactions. And we'll be getting into that in a bit or next week. So when you have, you see the sort of shorthand ATP goes to ADP. What we mean is this phosphate has been popped off and is going somewhere, being attached somewhere or carrying its negative charge somewhere. And then you're left with the adenosine diphosphate. When you incorporate a nucleotide into a DNA strand, you pop off 2 phosphates, and you're left with that guy. So we have 3 phosphates. Next, we've got the hydroxyl groups down here that are important. So this is a sugar, and remember, sugar's got lots of OH groups on them. When we draw the sugar with 2 OH groups, this is a ribose sugar. That's what a 5-carbon sugar is called, a ribose sugar. With 2 hydroxyls on it, we've got ribose. And this is the form of the sugar in ribonucleic acid, in RNA. Now, what we can do is actually pop that hydroxyl off and just have a hydrogen there. When we do that, we have made a deoxygenated form, or a deoxyribose. And so we've gone from RNA, ribonucleic acid, to DNA, deoxyribonucleic acid. In terms of chemistry, the loss of that OH group means that DNA is actually a bit sensitive to acids. There can be an acid attack there that can cause this thing to actually break apart. RNA is not as sensitive to acid. Other than that, they're very, very similar molecules, and we can substitute RNAs into our genome, and then they get pulled out and repaired all the time. So having a little bit of RNA in your DNA is not a big issue. We rarely have, if ever, DNA nucleotides in your RNA strands, but in terms of manipulation and the way we think about them, they're basically the same thing. One thing that, because of the enzymatic systems that exist with us and everything around us, RNAs end up being way more labile or delicate molecules. They are subject to being degraded really fast. Things like they're called enzymes that are called RNases, and RNases are everywhere. I mean, they're in the oil of your skin. They're, if you touch like a pipette tip and then use that to monkey around with RNA, you'll degrade the RNA very, very quickly. RNA isolation is a pretty involved process. It requires a huge amount of very strong denaturants because the RNA protein, or the RNase protein, is really tough. I mean, you can boil it and that'll stop it, but as soon as the temperature cools down, RNase is known being unique in that it can fold back up and get back to work, whereas most proteins, once you boil them, they're done. So RNases are everywhere and they're very potent. And this is just as a little interesting side note, the standard test for COVID-19, because it is an RNA virus, the test requires isolation of viral RNA, which is like a massive pain in the butt. We isolate DNA pretty easily. We can isolate DNA from ourselves by scraping a little bit of your cheek cells or spitting into a tube and just boiling it, and you liberate the DNA. To isolate the RNA, you actually have to do a good bit more work. And RNAs are really very delicate molecules. Consequently, the two vaccines that are currently in the U.S., the Moderna vaccine and the Pfizer vaccine, are RNA-based vaccines. Both of them are complexed up with some lipids that keep them a bit more stable, but you may or may not have heard, the Pfizer vaccine has to be kept at an ultra-cold freezer at minus 70 temperatures, whereas the Moderna is a bit more stable. You can leave it out actually for a while. Pfizer is a little bit like the Dippin' Dots of ice cream, and Moderna is a bit tougher. And I only mention that is, if you ever notice Dippin' Dots are not sold in the same case as your regular ice creams. Dippin' Dots, because they're little dots. Do we know Dippin' Dots? You never know. You always have to have a special freezer for them. I don't know how cold they are, but they are definitely colder than your standard freezer. Anyway, there you go. So RNAs are pretty labile molecules. All right. We can talk more about vaccines and RNA uptake and things. Actually, when we start getting into some of the stuff about transcription and translation, we'll probably throw a little bit of that in there. Okay, so one last bit here. When we're talking about nucleic acids, we can number the carbons, and the nitrogen base, for whatever reason, the nitrogen-containing base gets the straight numbers. The sugar gets the prime numbers. And so we have the ability to number the carbons. We got the 1' number, 1' carbon, 2' carbon, 3' carbon, 4' carbon, and then there's the 5' carbon right there. This notion, just as, the way I drew it, there's a carbon that attaches to that phosphate up there. That's the 5' carbon. This nomenclature is critical because we always talk about nucleic acid polymerization in terms of the carbons, and that we have this standard mantra of the 5' phosphate and the 3' hydroxyl. The 3' hydroxyl. And these are critical in terms of orienting the DNA sequence, the RNA sequence. So the 5' phosphate is always at the front end, the 3' hydroxyl is always at the back end. just like when we were building those amino acids, we had the amino terminus and the carboxy terminus. Now we're going to have a nucleic acid chain that'll have a 5' end and a 3' end. And that becomes really critical in all kinds of orientation things. You start thinking about nucleic acid polymerization and RNA and DNA interactions in general. So you've got to have that 3' hydroxyl, because when we link together two nucleotides, we're going to do it through what's called a phosphodiester backbone. And it looks like this. We got the phosphate groups over here, and OH, and we'll just make this one a ribonucleotide, so there's an H there. And then here's the base coming out, whatever it's going to be. So what we're going to do is pop off 2 phosphates. So they come out here. And we're going to make a link between one of the oxygens on the phosphate and this oxygen here. And this is known as the phosphodiester bond. And it's a pretty tough covalent bond as well. So that's how we start linking together nucleotides. And this process is known as polymerization. We're going to make a nucleotide chain using DNA polymerase or RNA polymerase. But we're always adding on to the hydroxyl group at the back end. So whoever is line leader, they stay the front, and you always add to the back. Just as another little final point, we were drawing here a deoxyribo. You can artificially synthesize something that's called a di-deoxynucleotide. A DDN. This actually is used a lot. Well, we use it in DNA sequencing all the time. It's referred to as a chain terminator. If you have no OH here, you can't add any more nucleotides to that end. Now, this is not a naturally occurring molecule, I don't think, but it is synthetically generated. And so dideoxys are used a lot for DNA sequencing, but also as an antiretroviral drug. So people that have HIV will be on a drug called dideoxyinosine. Inosine is a base modification that is essentially acts as a wild card. Inosine can bind or hybridize with either an A or a T or a G or a C. It can base pair with any of them. And so you throw in dideoxyinosine into a patient, and the way it works is that the HIV is a what's called a retrovirus, where it comes in as an RNA and it has to be converted into DNA, which is really very, very strange. We don't have systems in our bodies that do that. And the enzyme, known as reverse transcriptase, is not particularly picky about the nucleotides it puts in, and so it'll actually put in a dideoxy at a much higher frequency than our standard DNA polymerase will. And so we don't end up in the DNA in the nucleus pulling up dideoxys, but the viral polymerase does. And every time you put in a dideoxy, you stop the polymerization. Just a little point. All right, so that's some of the structural stuff. Let's think about making a long chain and see what happens. So this is some of the stuff that was going on after the war, or after World War II. A lot of work was being done in deciphering nucleic acids. One of the things I think is interesting in terms of like the history of molecular biology and the history of genetics is that Mendel, at the turn of the 20th century into the 19th century, his work, I guess, had been lost and rediscovered, and so the notion of genetics is growing. All right, it takes up until the early 1900s that some really fundamental experiments are being done to say what's known as the chromosomal basis of inheritance, because we have Mendel's work, but nobody knows exactly what the molecule is that is carrying this information. And it was probably assumed then there would be proteins. In fact, the guy, Thomas Morgan, who was sort of noted with the discovery of the chromosomal basis of inheritance using fruit flies, if you asked him, he probably would say, the chromosome is, yes, made up of nucleic acid. It's probably like a scaffold that holds a bunch of different proteins on it, because all we know is proteins are the things that actually do stuff. This nucleic acid crap, it's like starch, and I don't think it does anything. Okay, so it takes a lot of experiments, that we'll be getting into some of them, to actually get into the real notion that, somehow there is information in DNA and RNA that's actually being translated into producing proteins. We don't quite know what that is, but we do know that DNA has some information in it. So, one of the first things that was discovered and realized is that a guy named Chargaff, hope this will work after all that work, Chargaff developed his rules, known as Chargaff's rules. All right. This rule stated that if you take an organism, crack open its nucleotide, its DNA, isolate its DNA, which is pretty easy to do, and he could chemically determine the amount of adenine, guanine, cytosine, and thymine. What he found was the amount of adenine is always equivalent to the amount of thymine. The amount of guanine is always equivalent to the amount of cytosine. So not the hydrogen bonds here, but the actual equal sign. All right. So, that's an interesting notion. And things like X-ray diffraction and begins leading to the structure of the double helix. So if you could have gone back into Cambridge back in the early 1950s and found Watson and Crick and whispered to them, hey fella, the bases go on the inside, they would have to let you share the Nobel Prize with them. Because if we think about this, all right, so we've got these different macromolecules. Carbohydrates are just a glucose polymer. There's nothing but glucose. Lipids, it's just long-chain hydrocarbons. Proteins, now there you have something. We got these 20 different amino acids. We got all these structures and shapes. That's something that can do something. But nucleotides, nucleic acids, they look a lot like starch. I mean, it's all negatively charged. It's just this long-chain polymer, and it doesn't really fold up into really cool shapes that can do stuff. I don't see how it has any information in it. All right, so the ATGC business, we have to think, well, maybe there's something there. The idea would be, all right, the base pairs are going to be sticking out like this, and you're going to have this phosphate core in the middle, and that way some kind of machinery can read through and look at the bases. Okay, maybe. So they work to try and build a model that way, and then finally they go, what if we turn it around? And now all of a sudden, you have the reason for Chargaff's rules, because you've got this double-stranded molecule in which you've got A base pairing with T, G base pairing with C, and it just goes on and on and on as the DNA sequence. And, it's like, eureka, this actually works. It makes sense. There's some pretty cool implications behind this. This must be how it works, in that we have a double-stranded molecule. So we've got two nucleic acid molecules running in opposite directions. So we have the 5' end here to the 3' end here, and the other strand is running in the opposite direction, from the 3' to 5' direction. And then we have the bases in the middle. So we got A, T, G, C, A, T, A, T, G, G, Z, 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 G. Blah, blah, blah. All right. And they're heroes. Now, what they see is based on a lot of different structures and things. This thing actually does have the ability, and because of the way the charges are interacting, it's going to twist and form this double helix. And so whenever we're drawing DNA sequence, unless you're like a super duper artist or you really want to be confusing, nobody ever draws it as the helical structure. And I will now attempt to do it. Whatever. What we got here not bad. Think of a step ladder that a giant, or a extension ladder, aluminum extension ladder, that a giant grabs and goes and twists. There's actually a Cold Spring Harbor Labs, which is a part of State University of New York, Stony Brook, out on Long Island, is a institution that Jim Watson was the head of for a very long time. I'm not sure if he's still alive. God. Anyway, Cold Spring Harbor is like a campus devoted to DNA, gene expression, and molecular genetics. Very cool place. Everywhere you go, there is homage to the structure of DNA. There's like wallpaper strips that are three-dimensional models of DNA molecules. Just everywhere you go, there's a DNA-based nucleic acid motif. And they've got in the middle of the campus a gigantic but drawn to angle scale structure of a double helix. And it's an interesting thing to think about. So you can imagine what this thing, it looks like a spiral staircase without handrails. And when you're walking up this spiral staircase, you're actually stepping on the hydrogen bonds. So watch your step there. And the sides of the staircase are the sugar phosphate backbone. Additionally, when we think about this, we have this sort of topological terminology of the space where your body is referred to as the major groove, and the space that your feet are in is referred to as the minor groove. And we'll kind of get back into this when we start talking about DNA sequences and stuff. But it's some very interesting topology. That said, nobody draws DNA, when you're trying to think about sequence, as a double helix. Because there's going to be an exam question where I'll say diagram a molecule of DNA, and somebody will invariably draw it as a spiral structure. And no, you can't keep up with which side is which. So no, we always draw DNA, sadly, as just a little stripe. But there you go. So that's nucleic acids. Now, one final point. Oh, I got 5 minutes. We got here now a very complex molecule. When we have double-stranded DNA, it can fold up into these helical structures, and there's different sorts of helices, and that's fine. But single-stranded RNA is an interesting molecule in that it has the ability to form intramolecular base pairs. So we were talking about intramolecular hydrogen bonds. We can have base pair complementarity within an RNA molecule. So a single-stranded RNA molecule might have the ability to form structures like this, what's called a stem and loop structure. As it turns out, that's what a tRNA looks like. And so you'll have base pairing here, and base pairing here, base pairing here. And what's really cool is you unzip this thing. You're just looking at it as a linear sequence. You can identify spots that are going to have complementarity. So you might have a whole stretch of GC, GCC over here, and then running on this side, you'd have a GGC, GC, and you go, and these are in two way different parts of the RNA, but you can scan through it, or have your computer do it, and you can see, that's a spot that's going to form secondary structure. And RNA molecules have the ability to form up into very complicated secondary and even tertiary structures just through base pairing. And not surprisingly then, when you get an RNA molecule that can fold itself up into some kind of very distinct shape, it can actually have some enzymatic activity because we've got a really complex structure here. And this is a guy named Phil Chet, back in the 70s, was the first to discover this. What are known as RNA enzymes are what he calls a ribozyme. And these are RNA molecules that fold up into shapes that actually have catalytic activity. Now, sadly, about the only thing ribozymes do is cut other RNA molecules or cut DNA molecules. They don't do anything all that amazingly cool. I mean, that's pretty cool, but it's like, it would be neat if you could get an RNA enzyme that would actually, like, break down a sugar or something. It doesn't work that way, but it does hold and kind of reinforce this notion that secondary and tertiary structure is required for complicated biochemistry and catalytic ability. And things like lipids and sugars, they don't have any kind of secondary and tertiary structure. Consequently, they can't really do anything. They're just, they make pudding, so you don't really get great structures. All right, so we will kind of take some of this stuff in terms of the directionality and base pairing when we start talking about transcription and translation in another week or so, 2 weeks, and carry on from there. And, all right, I think, that gets us through the major macromolecules of living systems. B, we're on to energy and enzymatics. And C, that's all I have to say for today. Have a lovely weekend. On Monday, we're all out of quarantine or soft quarantine or something, and God knows what that means. But we're not doing lab for a while, sorry. And that's all I know. Is there anything else I'm missing? Perhaps not. All right, I'm going to turn the recorder off now. Bye-bye.